So how do we get our traits? If you have blue eyes or brown eyes, how did that happen? Well, most of us understand that our genes or our DNA actually determine our traits. But what's this process that goes from some chemical in our cells to actually getting blue eyes or brown eyes? Well, in this video, that's what we're going to investigate. How the genes in our DNA get translated into the proteins that make up our traits. Let's get started. Okay, so I want you to think about where your DNA is held. Think about that. Where is the DNA in my body? Well, most of it, you may remember, is in the nucleus, right? So here's the nucleus in a cell, and here's the DNA. Now, I'm going to give you sort of an analogy for this. It might seem a little complicated, but I hope it will help you with this process. If you think about it, your DNA is sort of like a recipe book. It's a recipe book that's got all the recipes to make your traits. Blue eyes, brown eyes, red hair, certain enzymes to be made, right? The DNA is really just like a recipe book. Now, when we want to make a certain trait, right? Let's talk about all the genes, for example, that make blue eyes. Those genes are held tightly in the DNA in the nucleus, but they have to be transcribed into messenger RNA. So in my analogy, let's say we wanted to make a certain recipe. So I go over to my DNA, it's the recipe book, but I don't want to make every single recipe in the book, just like I don't want to make every single protein that's in my DNA. Well, so the body picks which recipe it needs, right? So it takes a few recipes and makes a copy of them. We call that process transcription. That's the process of taking a little bit of DNA, and making it into messenger RNA. So it's called messenger RNA because it's the recipe, it's carrying the message. Now, where does that go? Well, all good recipes need a chef, right? To mix the ingredients together, put them in the correct order. Well, the chef in our body is actually the ribosome. So let me show you what's going on here. If you think of the ribosome as our chef, right? That is what is going to ultimately make our protein. And remember, our proteins, these series of amino acids, make our traits, like eye color. Well, this is actually quite a bit more complicated, right? There's a lot going on down here. The chef, the ribosome, needs all of the ingredients to make the meal, to make the dish, to make the protein. So what happens is, through the process of translation, right, messenger RNA, the recipe, goes to the ribosome. The ribosome, also known as the chef, puts the ingredients in the correct order. Now, how do the ingredients get there? They come over on some transfer RNA. Transfer RNA brings the ingredients. In this case, the ingredients are amino acids. All right, so my analogy here is pretty detailed. I want to go through it one more time to make sure you understand what we're going to talk about. We're talking about how do we get from the genes in our DNA to our traits. How does this happen? Well, the recipe book, the DNA is wrapped up tight in the nucleus. So instead of bringing the DNA out, we just make a nice little copy of the recipe we need. We don't need to copy the whole recipe book. We just need one particular gene to be copied. So that's called messenger RNA, that one specific gene that's going to be copied and that we need the recipe for. Then that messenger RNA will go over to the chef, the ribosome. Remember, the point of ribosomes is to make proteins. So where does it get the ingredients to make the protein? Transfer RNA brings the amino acids over to the ribosome so that the chef can put them in the correct order. Once we get the right protein, well, that is our phenotypic trait. The proteins are what make us what we are. So if I have brown eyes, that's because the proteins in my eyes reflect brown light. Okay, so this is what we're going to talk about in this video, this process of translation right here. Okay, so how does messenger RNA code for proteins? Well, let's start again with our DNA. Remember, we've got double-stranded DNA. So what happens is, through the process of transcription, which we learned about in the last video, DNA codes for messenger RNA. So if this was our original coding strand, right, this is the template for this. So notice A goes with U, T goes with A, etc. Here's our messenger RNA being made. Now, that makes sense because really the nucleotides are just lining up with their counterpart. But how do we go from this, which is all nucleotides, to proteins, which are made of amino acids? Well, we use a system called codons. And basically, it's a sort of a three-letter system. These three letters, these make up a codon. Each of these codons code for a particular amino acid. So UAC codes for tyrosine. UCA codes for serine. GGU codes for glycine. UCA codes for serine. Then this order of amino acids make up the protein. Okay? So messenger RNA codes for protein with the help of codons, a three-letter system to code for a particular amino acid. All right, so let's look a little deeper into these codons. So here's the definition. A codon is a block of 3 nucleotides that decodes into one amino acid. So like I mentioned before, if we're looking at the messenger RNA, notice that UAC codes for tyrosine. Now, I want to point out a few more things. Remember, when DNA is being read, we read from the 3 to 5 prime direction. So in the process of transcription, DNA is read from the 3 to 5. But now notice this. The messenger RNA that was made from this up here is in the 5 to 3 prime direction. So translation, we read the messenger RNA from the 5 prime to the 3 prime direction. That is supposed to be a 3. Okay, that's what a codon does. So then what you'll notice is I've just been writing, right, using little circles to show this amino acid. But remember, these are actually chemicals, right? There's tyrosine, there's serine, there's glycine, there's serine again. And remember, when these connect, right, they make these peptide bonds we learned about quite a bit ago when we were talking about how proteins are made through dehydration synthesis, right? These connect, and it's basically making one long series of amino acids linked together, and those are polypeptides, which ultimately make proteins. Okay? All right, so let's talk a little bit more about the code. Well, this is fascinating to me because what we find is that these codons that code for amino acids actually code for all life, right? This is one of the strongest supporting pieces of evidence we have that life originated from a common ancestor, right? Because whether you're a plant, an insect, a human, we all have the same code in terms of codons coding for particular amino acids. So here's a example of a codon chart, right? So if you look in this chart, let me tell you how to read it. Remember, we always read from the 5 prime to 3 prime direction when we're reading messenger RNA. All right, so this is the 5 prime, and then we go to the 3 prime over here. So let's say that you got in your messenger RNA sequence, you had AUG. Well, how would you know what that was? Well, you start over here, A, and then U, and then you come right here to G, methionine. AUG is methionine, which also happens to be the start codon. This is what we see at the beginning of all of our polypeptide chains. Now, there are also some stop codons. So when the ribosome reads UGA, all right, let's find it. UGA, there it is. That's a stop codon. UAA, UAG, these are all stop codons. That signals to the ribosome, stop making this particular protein. Okay? So there are several types of these charts, and you will probably be asked to decode a particular piece of DNA into RNA and then into the amino acid sequence to tell me what our proteins are. So I want to show you this other alternative code of life chart. I'm not personally a big fan of this. I think this is misleading. And let me show you why I think that. But I want you to understand it in case you see it. So let's say we have AUG, the start codon. Well, if we start here. Okay, A, we start in the middle and then we go out. A, oh, I don't see a U because they've replaced that with T. They're using the DNA nucleotide instead of U in this particular chart, right? It's still AUG, except they're using the T because this is one of only the DNA, not the messenger RNA, okay? It's rare that you would see a chart like this. Hopefully it will have the correct messenger RNA in there, but sometimes they put the DNA nucleotide instead of the RNA nucleotide in here. But you would do it just the same. So let's do one that's a little easier. Let's say CCG. So C goes to C, goes to G. You know that's proline, okay? The only difference is that this one uses the T instead of the U. Hopefully you won't run into many charts like this. Okay, so how do the codons get matched to the amino acids? Because I say, all right, well, this particular codon matches to this amino acid. Well, the answer is with anticodons. So let me explain what that means. All right, so remember, when DNA is transcribed, we go from DNA to messenger RNA, right? All right, so here's our messenger RNA. Remember, a group of 3 of those is called codons. All right, so UAC codes for something, but how does it get there? How does the amino acid actually get there? Well, it's brought over, if you remember my very first slide, by transfer RNA, right? That's what's going to bring over the amino acid. So we've got these codons down here, and if you notice, when this right here, this is a transfer RNA, that's not great writing, transfer RNA. What you see there is that on the bottom of transfer RNA is the anticodon that corresponds to this codon. So what goes with U? A. What goes with A? U. What goes with C? G. So the anticodon bonds to the codon. That gives the signal that this amino acid that is attached to this transfer RNA is the correct one. In this case, it happens to be tyrosine. Okay? Now, I did this one for a reason. Notice tyrosine is not the start. This is not the start codon. This is the anticodon that goes to tyrosine. If this was AUG, the start codon, the anticodon would be this, and the amino acid attached would be methionine. All right, so let me show you another example. If serine was the next amino acid in the sequence, the anticodon on the bottom of the transfer RNA would be AGU, because that corresponds to UCA on the codon. So remember, anticodons are on transfer RNA. Codons are on messenger RNA. These are different. Codons on the messenger RNA, anticodons on the transfer RNA. Anticodon matches the codon so that we know the correct sequence of the amino acids being brought by the transfer RNA. Okay? Oh, I've labeled it there for you, transfer RNA. Then, obviously, when it's all complete, we've got the tyrosine, serine, glycine, serine. We've got our correct amino acid sequence. How did the amino acids get there? Transfer RNA brought them, matched the codon, so that we get the correct sequence. Okay, so let's talk a little bit more about this tRNA. We say it has a clover leaf structure. So here's the example I used earlier of what you might commonly see it as sort of just like a little icon. But actually, if you look at the structure of the RNA, you see it sort of forms this clover leaf pattern. And what you find down here is the is the anticodon. So we have a 3' and a 5' direction. 3' is here, right? It goes all the way around, loops, 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 and then back here to the 5' end. Because remember, DNA and RNA have ends, 3' and 5'. This end up here is the amino acid attachment site. So this is where the amino acid attaches, and this is the same thing in a different looking diagram. On the bottom is the anticodon. So in this case, this little purple region down here is actually the sequence, the AGU, right? AGU, those particular nucleotides in that sequence, that is where the anticodon is, okay? So tRNA, cloverleaf structure, that's sort of what it looks like. Now, how does the tRNA actually have the right amino acid, right? We're saying the tRNA is transferring down the amino acid, but how did that amino acid even get on that tRNA? Well, there's an enzyme called aminoacyl tRNA synthetase. This enzyme bonds the amino acid to the transfer RNA. So here's the enzyme. What happens is you'll see that the transfer RNA is down here, the amino acid is here. So this enzyme helps those two bind. Another example would be here. This is a little more easy to see. Here's our transfer RNA, anticodon, amino acid attachment site. Here's the particular amino acid that gets joined to that transfer RNA by this enzyme, aminoacyl tRNA synthetase. Now that sounds like a lot. That's a mouthful, Herder. Okay, fine. But if you think about what it's doing, this enzyme makes sense. What's it doing? It's bonding the amino acid to the transfer RNA, and it's sort of synthesizing that whole mechanism. So therefore, it's aminoacyl tRNA synthetase, putting them together. All right, so what does the ribosome do, right? We've known since the beginning of the biology course that ribosomes make proteins, right? When you say, what does a ribosome do? It makes proteins. Well, it's a little more complicated than that, right? What a ribosome really does is facilitates the coupling of the tRNA anticodon to the messenger RNA codon. If those match, then it gives its amino acid to make that protein. All right, so what's a ribosome made of? Well, ribosomes are actually made of ribosomal RNA. Notice this is the third type of RNA we've talked about in this video. We talked about messenger RNA that has the code. We've talked about transfer RNA that transfers the amino acid down. And now we're talking about ribosomal RNA, which makes up the ribosome. And then, of course, there's also some proteins in that ribosome. We also know that ribosomes have two subunits, a large and a small. So here's an example, sort of a 3D dimension, right? Here's the large subunit and then the small, right? Large and small. You might also see it represented like this, with the large subunit here and the small subunit here. These three things here are the attachment sites and the exit sites. So when the transfer RNA comes in, it attaches, moves through here, and then exits. We'll go over that in just a moment. And then here is what you might also see of the ribosome. You'll notice here that we've got the ribosome. This down here is the messenger RNA, right? Here's the transfer RNA. Here's the anticodon. Here's the codon. And up here is the amino acid that's going to be joining to make this amino acid chain. Well, what do we call a bunch of amino acids together? A protein or a polypeptide, okay? So what is the ribosome doing? It's allowing this to occur so that we can make this protein up here. All right, building a polypeptide. Well, there are three steps: initiation, elongation, termination. Well, these make sense too. Initiation, we need to initiate the whole thing. So what we need to do is we need the recipe, we need the code, the messenger RNA. We also need the chef, the ribosome, and some other initiating transfer RNA. This is all in the initiation stage. So the first thing that happens, notice that the large and small subunits of the ribosomes are apart. But when this messenger RNA arrives, when we get the recipe, it's now time to make the dessert, right? Time to make the protein. So here's our recipe. And then what we see is when we get to the start codon, AUG, we get the transfer RNA that comes and binds. This initiates the whole complex now. And we have our first amino acid, methionine. Okay, well, that's the initiation. Next will be elongation, step 2. So this is when our polypeptide chain or our protein chain is growing as we add new amino acids, right? So notice a few things here. Notice that this was our first one, right? There's our methionine amino acid. Then it was followed by this transfer RNA that had the anticodon that matched this codon, and it brought a different amino acid. Now, think of this as a moving target, right? They come in, bond, give the amino acid, and then they leave. So each of these sites actually has a name, and I'll go over this more in a moment, but just so that you know, this right here where the transfer RNA attaches is the A site. This site right here where the polypeptide is growing is the P site. And then this site here where the transfer RNA exits is the E site, all right? So the transfer RNA comes, it matches, it comes to this stage, donates its amino acid, moves to this stage, exits. This is essentially the elongation stage where we're getting longer and longer polypeptide chain. And then finally we arrive at the termination stage. So we need a release factor. Remember those release proteins that are bound to the A site will essentially release the whole thing. We get there when we get one of those stop codons. So basically, right, when we get the stop codon in place, right, this signals for termination. So we have this nice polypeptide that's been growing, but now we've got the stop codon that's being read, which tells this transfer RNA to bind, which then, as we see, we have this release protein binding. Now the whole thing is released, and we have our nice protein that's being made. What protein is that? Well, it doesn't know. Maybe it's one of the proteins that contributes to your eye color or hair color, or maybe it's making an enzyme for you. Okay, I'd love to put this all together, and then we're going to start to wrap things up with a bit about mutations. So let's connect all the dots. We're going from gene to protein. Where are our genes? In the nucleus on our DNA, right? So here's our DNA. And then we have an enzyme here that looks like it's making messenger RNA. Do you remember from the last video what that enzyme is? RNA polymerase. So RNA polymerase makes the messenger RNA. Now, here's our messenger RNA, but if you remember from the last video, we needed to do a few things. We need to add a 5' cap, a poly A tail. We also need to remove the introns and leave the exons. We leave the exons because they're expressed. We remove the introns because they're in between. So now, once again, you see we've added the 5' cap, the poly A tail. We have removed the introns, so now we're ready to leave. This messenger RNA is now mature, and it's going to go over to our ribosome. Oh, there it is. Goes to our ribosome. We've got the large subunit. We've got the small subunit. Then, remember, part B of the story is taking place over here. I've got a bunch of amino acids. These amino acids will be facilitated, their coupling to the transfer RNA by aminoacyl tRNA synthetase. So this enzyme takes the amino acid, joins it to the transfer RNA, and then it sends it on its way. Remember, these two things are the same thing. They're just two different ways of showing it. That transfer RNA is now ready to go to the ribosome if it's needed. How will we know if it's needed? Well, remember, the anticodon on the bottom of that must match the codon on the ribosome. So here we go. We see that this anticodon matches the codon there, so it's going to fit right into this A site or attachment site. This was our mature messenger RNA that is being read. Now remember, messenger RNA is read from the 5' to 3' direction by the ribosome. All right, so we have the attachment site, we have the polypeptide site, we have the exit site, and then this is our growing protein that we're making there. Okay? Very complicated, yes. But basically, what's at the root of this? Well, the root is that it's how I started the whole video. You got DNA. We want to make proteins that are our traits, blue eyes, brown eyes, red hair. How does that happen? Well, this whole slide is how that happens, okay? From the nucleus to the ribosome with the help of transfer RNA, making these polypeptides. Okay, can things go wrong? Of course. Sometimes we have mutations. Well, I shouldn't say wrong because we also know that mutations help evolution. So can things be different? Yes, mutations. Mutations come in a few different forms. We have point mutations when we have a single point of DNA that has changed, that will change the amino acid. So let me give you some examples. So let's say we're here at our DNA level. This example over here is no mutation. Notice with no mutation, our DNA code is TTC. Well, if you think about it, what does T match with for messenger RNA? Well, T goes with A, T goes with A, C goes with G. So our messenger RNA will be AAG. If you go back to my chart and you look, AAG codes for lysine. And there, if you look, is the lysine amino acid. Now, there are a few different types of point mutations I want to talk about. Right, so here was no mutation, that's our original. If it's a silent mutation, there could be a mutation in the DNA that doesn't affect the protein level. So notice here, the C got turned into a T. So our new DNA is TTT, which codes for AAA. But remember, if you think back to our chart, the code is redundant. AAA and AAG both code for lysine. So because they're both coding for the same amino acid, we call this a silent mutation because it really didn't change the amino acid. It was silent in its effect. Okay, a nonsense mutation, I like this one, it's just nonsense, because what it does is it's a mutation that codes for a stop codon, right? So instead of TTC, we had a mutation at the first one that coded for an A. A goes to U, T goes to A, C goes to G. UAG codes for stop. That means the protein is stopped being made. Okay? Then there's two types of missense. This is where we get the wrong amino acid. A conservative missense, in this case TCC, codes for AGG, coding for arginine. That's different, but notice the properties of arginine are similar to the properties of lysine. So it's conservative because the properties of this amino acid are close to that, doesn't make that much effect. But if you look at a non-conservative, that coded for the codon ACG, which codes for threonine. That is very different from that. So we're going to have a complete change in the protein function because of the interactions of those chemicals. All right, last slide talks about when we have a mutation that shifts the whole frame. What do I mean? Well, instead of a replacement, right, this nucleotide is replaced with this one, we actually either insert one or delete one. So let me show you how this works. Here's my wild type of messenger RNA, right? This is what we would say is our normal sequence without any mutation. What if there was a deletion? So if you look right here, what if this U was just deleted, taken out? Well, no U, then everything shifts down the frame, right? It's like when if you were taking a test with a Scantron and you skip a question, right? And you bubble the next one up where it should have been, and now everything after is wrong, and you got to go back and erase everything. Well, that's sort of what's happening here. If you miss one, we take one out, then everything downstream is wrong. So we're getting the wrong amino acids and thus the wrong protein being made. Well, the same thing occurs if we do an insertion. If we have an extra added, right? In this case, it coded for a stop codon, but it may just shift everything where all these resulting amino acids are also wrong. Okay? So we've got point mutations, which change a single point, right, for one for another. And then we have frame shift mutations, which change the whole sequence down the frame. All right, hopefully this video helped you understand how we go from DNA to traits or from DNA to protein.